Assalamualaikum to our lecturer, Madam Dia. I'm the first presenter, NURUL AYUNIE binti ADZHAR from RAS 1154A. I will present our video presentation task. UVVs, or referred as ultraviolet spectroscopy, refers to absorption spectroscopy or reflectance spectroscopy in part of the ultraviolet and the full adjacent visible spectral regions. Next is DNA is the hereditary material in humans and almost all other organisms. Next, I will explain the importance that we get from analyze the sample from the three articles that we choose. The first article is recovers about comment on the frequently used method of the metal complex DNA binding, constant determination from UVVs data. The importance that we get from analyze the sample, which is we can know the changes in the UVV spectra of the mixture containing metal complex, and DNA are caused by partial dissociation of the coordination compound and completion of the released copper to ions with DNA. In addition, the partial dissociation of the metal complex in presence of DNA from the signals in the EPR spectra assigned to dehydrated copper to ions. Next, we move to the second article, which is recover about determination of DNA quality and quantity using UVV spectroscopy. The importance that we get from this article is we can determine the purity of protein and DNA. The second is the ratio of the absorbance can be determined to assay the DNA quality and quantity for the previously extracted samples when we use the absorbance of a DNA sample at 260 nanometer and 280 nanometer. Lastly, the third article that we choose recovers about UV visible spectroscopic effect on hemoglobin and DNA degradation. The importance that we get from this article is to maintaining the integrity of the forensic evidence. The real problems lies with a systemic failure that does not properly account for evidence from collection through final disposal. There are three main types of blood cell and cell fragments, which is red blood cell, white blood cell, and platelets. Red blood cells, also known as RBC, do not have a nucleus and thus no DNA. The main component of RBC, which is red blood cells, is a protein that contains iron and is responsible for oxygen transport the hemoglobin. Hemoglobin, also known as Hb, is the main component required in forensic serology. Before the advent of DNA typing, the ABO system of antigens in humans erythrocytes that contain a variety of blood group antigens in the most frequently used blood group system for forensic community. Lastly, for me, the precision of the instrument to DNA sample is quite good. For example, in forensic cases, in that cases we can determine the component that has in the evidence. The well-preserved evidence will exist in linking someone to or excluding someone from crime scene evidence.
Assalamualaikum warahmatullahi wabarakatuh to Madam Dia Shaliana. My name is SITI AIFA NASUHA binti DESA and I'm from RAS 1154A, and I will explain on how UV ultraviolet visible spectroscopy works and how they operate. From the article about DNA, When light, either visible or ultraviolet, is absorbed by valence or outer electrons, these electrons are promoted from their normal states to higher energy excited states. The energies of the orbitals involved in electronic transitions have fixed value. Because energy is quantized, it seems safe to assume that absorption peaks in a UV visible spectrum will be sharp peaks. However, this is rarely if observed. Instead, the spectrum has broad peaks. There are also vibrational and rotational energy levels available to absorbing materials. Light absorption can occur over a wide range. A light from 190 nanometer to 900 nanometer is usually used. Valence electrons are found in three types of electron orbitals. Sigma bonding tend to be lower in energy than pi bonding orbitals, which in turn are lower in energy than non-bonding orbitals. Most of the transitions from bonding orbitals are too high a frequency. The exact energy differences between orbital varies. In organic molecules, double bonds which are next to each other can conjugate, joining together and delocalize the electrons over all of the atoms. As a consequence, molecules with many conjugate double bonds can be colored because they absorb energy in the visible as well as the ultraviolet part of the spectrum. Now I will explain the component in instrument for optical absorption measurement in UV-Vis, which is a spectrometer, a spectroscopic instrument that uses a monochromator, polychromator in conjunction with transducer to convert radiant energy to electrical signal, and the spectrophotometer is spectrometer that allow measurement of the ratio of the radiant powers of two beams that can measure absorbance. Spectrometers employ a grating or a prism monochromator to provide a narrow band of radiation for measurements that the wavelength used can be varied continuously, thus making it possible to record entire absorption spectra. The function of photometers is use an absorption filter or an interference filter. Next, there are two types of beam in ultraviolet visible spectrophotometer, which is single beam instrument and double beam instruments. In single beam instrument that used in the visible region of the spectrum, simple and inexpensive spectrophotometer. The single beam instrument can be found in Spectronic 20. And the second one is double beam instruments, which is a modern photometers and spectrophotometers. There are two types of double beam instruments, which is double beam in space and double beam in time. The double beam in space is that has a V-shaped mirror called beam splitters that can split the radiation from monochromator and can be filtered to two beam. The disadvantage for double beam in space is difficulty in matching the two detectors. Then, the double beam in time. Double beam in time, the beams are separated in time by rotating sector mirror that directs the entire beam through the reference cell and then the sample cell. The advantage in using double beam is compensate the most short-term fluctuation in the radiant output of the source, then compensate for wide variation of source intensity with wavelength.
Assalamualaikum to Madam Dia. My name is NURUL binti AHMAD, and I will be continue talking about why UV spectroscopy are chosen in determination of DNA. So, based on the article of determination of quantity and quality of DNA using UV spectroscopy, both of it have protein and DNA. So that is the reason why they are using the UV spectroscopy, because protein and DNA have substantial absorbance in the UV region. Also, it can be used to determine the purity of protein and DNA. And UV spectroscopy have many advantages, such as they are widely available, consumable, and doesn't require any specialist reagent, and also they find the UV spectroscopy method is very quick. Based on the second article that I have read, which is the effects of hemoglobin and DNA using UV spectroscopy, they use UV spectroscopy because they want to determine the preservation related color changes in human blood. So basically, in blood, we have white blood cells, and inside white blood cells, there are two things, which is DNA and hemoglobin. So they need to use UVVs for an edge determination of the blood. And basically, they will use all the DNA sample in UVV instrument to determine the preservation related changes. Means that they wanted to determine the color changes from red color to brown red color. And next is for the third article, which is the article is about the metal complex DNA binding constant determination from UVV's data. They use UVV's data to strict constant of binding between DNA and metal complex. And as we all know, UV spectroscopy is the only method to determine the qualitative and quantitative when to study of those interactions. From my opinion, based on the three articles, I think the article about the determination of quantity and quality of DNA is more accurate than the others, because they use the DNA sample to analyze with 260 nanometer and 280 nanometer absorbance values. By using the absorbance of DNA sample at 260 nanometer and 280 nanometer, the ratio of absorbance can be determined to assay the DNA quality and quantity for the sample. They have the exact values for the experiment. Also, when we are using the absorptivity, it can also be used to determine the concentration of sample using Beer-Lambert's law. And one more thing, the value is more accurate because we can only calculate it by using the formula. Why I think the hemoglobin and DNA article isn't accurate? Because based on the article, the method may not be suitable to be used on the specific human DNA. From the article, they basically use hemoglobin and DNA, and both of it are from human white blood cells. And they also suggesting other method that can be used for testing the DNA, which will works better than using the UV spectroscopy. And for the third article, why I think it is not accurate, because based on the article, they conclude that it said many researchers are suggesting the interaction between metal complex and DNA should be first demonstrated that the observed experimental data could not be explained without hypothesis. Also, the very existence of metal complex DNA could not be concluded only from the UV spectroscopic data. So, I think that the only accurate value for based on the three article is only the article of the determination of quantity and quality of DNA. Thank you.
So, Assalamualaikum warahmatullahi wabarakatuh. To Madam Dia Shaliana binti Mat Syukri. So, my name is Muhammad Huzaifah bin Yaacob as the last presenter of my lab group. And I want to make a presentation about the last elements, which is how the cited experimental data can be used to achieve the objective of the study, limitation and advantages of the method used, and the recoveries of the article. So first thing first, we have to know what is objective. So objective is what it's supposed to accomplish in experimental procedure itself, and the characteristic of research objective, it should be clear, complete, specific, and we have to identify the main variables of the study. And the most important thing when we are doing the experiment, that we have to take the results and data. So result and data is two different things. So first thing first, data is refers to the actual numbers or other information you obtain in our project. So data can be represented in tables or charts if appropriate, where the result is where the data has been manipulated or the hypothesis is tested, and it can be also shown in tables, charts, or graph. So the method that we have used is ultraviolet or visible spectrometer. So we have to know what is spectroscopy. So spectroscopy is the science concerned with the investigation and the measurement of spectra produced when materials interacts or emits electromagnetic radiation. So for the spectroscopic measurement, we have to use the interaction of radiation with matter to obtain information about sample. So sample is stimulated by applying energy in form of heat, electrical energy, light, particles, or chemical reaction. Specific types of interaction that we observe depend strongly on, first one is the energy of the radiation rose, and the second one is the mode of emission. So the limitation of the method used is we must clear the area of any outside light. Second, we must clear the electric noise. And the last one is we must clear other contaminants that could interfere with spectrometer's reading. And the advantages of the method used is it quick analysis ability. Second, easy to use. And the third, when using light to identify and characterize matter, light requires no physical contact between samples and the instrument. So for the last one is the recoveries. We have find the 3 articles. The first article is from Alkins 2013. It recovers about determination of DNA quality and quantity using UV spectroscopy. And the technique is non-destructive and can be used also to determine the purity of protein and DNA. For the article 2, which from Gamov 2019, it's recover about comment on the frequently used method of the metal complex DNA binding constant determination from UV data, and the result obtained by the analysis of a number of the reaction that could occur in the solution of copper to anions, buffer components, namely tris, ligand, hydrazone, and the Na itself. And for the last article, which is article 3 from Cow 2019, it recovers about the effect on hemoglobin and DNA degradation, and the samples are gathered from the period 2013 until 2018. And we can know that the degree of preservation relate changes that include first, changes in maximum absorbance of solid band, and second, changes in the wavelength of the 1 max for the solid band, which is for preservation of blood. So, as a conclusion, spectroscopy method is suitable used to quantify the concentration of the protein and DNA in a solution, and also can be used to analyze fluorescence from a sample in a form of absorption spectroscopy. So that's all from us. We hope that our topic can make us really help us in our daily life. So with that, I thank you.